will not forget to record. So it's now being recorded.
Excellent. Okay.
But we'll give them a few more minutes to show up.
That's fine. Totally fine.
What's the weather, Gayle, in Fort Collins?
It's really nice out today. I took a walk at lunchtime and I don't know what it is. It's probably definitely in the 40s, maybe even warmer. Sunny. It's nice.
Yay. Yay
Nothing
for
fast.
sun. We went biking this past weekend. So this is a photograph from where we were biking and about an hour drive south of here in Oregon. I think it looks so lush. It looks like some subtropical place.
It looks lovely. We're definitely not looking very lush out here right now.
Well, is snow still on the ground?
Yes. In fact, what, Saturday we had some more flurries come along. Yeah, we're still getting some snow. My front porch always has snow, so it'll last another probably month before that all melts off. But the roads are all fine, so we're good.
All right, 129. Robert Kruger's here. This is good. Connecting to audio. Oh, a few more are connecting.
Hi, Gayle. I just wanted to say hi really fast.
Hey, Jenny.
Good to see you.
Nice to see you too.
Hey there, Jenny. Good to see you. We have the appointed hour, but not too many participants yet.
You know, that's okay. It'll all be recorded and we can make them available elsewhere.
Yeah. I'll give them, 30 seconds.
Okay.
Yeah, a few more folks signing up.
Well, our building's tricky right now. I don't think we have network access at the building at the moment. We didn't all last week. So,
well, yeah, last week I was trying to manage this call and I found out it was because you guys had a flood out there somewhere. And of course my VPN goes through your location. And so I was not able to connect.
oh dear. Oh, so that's way bigger than just local. That's too bad.
Oh yeah, but at the moment I'm connected. This is good. So.
Okay.
Well, I bet we're going to have about, at least twice this number. But I suppose at this point, I would like to begin and introduce our speaker today for our clonal training sessions. We have Dr. Gayle Volk, who is a geneticist out at the NLGRP. And she's been working on cryogenic preservation of all of our crops. We're so happy that she's backing up citrus and apples and pears and all sorts of clonal material. And so she's going to give us at least talk about the clonal material that she's working with. And I guess I'll just turn it over to Gayle at this point. So Gayle, go ahead.
Thanks. And I'm gonna work on the sheet screen sharing thing. Let's see if this works. Okay. And you can see that?
Yes, everything's looking very good.
Great. Okay. Well, thank you for this opportunity. And I'm splitting my presentation into 3 parts today. And ideally, we'll take 2 of these parts and flip them into training materials and post them online at a later date. The first part is going to be a big picture overview of the vegetatively propagated collections in the gene bank system and their backup status. The second part talks about preservation of those clonally propagated plant materials at the National Laboratory for Genetic Resources Preservation here in Fort Collins. And it's not the nuts and bolts on how we freeze things. It's instead the flow process of how materials get to our facility, kind of the big picture of what happens to them, and then how you can access information about that. And the third part is about the online learning work we're doing and developing training resources for plant genetic resources management and use. So here comes the first one. Oh, hold on. I don't know how to advance the slides. Okay. So conserving vegetatively propagated collections in the USDA National Plant and Germplasm System. This is an exciting year to be giving this presentation because it's the International Year of Fruits and Vegetables. And many of our fruits and vegetables are clonally propagated. So it's particularly relevant for this training series that Kim is coordinating for. The vegetatively propagated collections are those that are in the big umbrella of things considered to be propagated by grafting or cuttings or some other method rather than grown from seeds. And within the National Plant Germplasm System, we maintain these collections in fields or in greenhouses and screenhouses or in tissue culture. Parts of these collections for some crops also reside as seeds or wild collected seeds in freezer storage as well. But today we're emphasizing the vegetatively propagated parts of those collections. These collections have really high economic value. They tend to provide a lot of nutrition to diets, they add variety, and they're also really important to local economies for regional or international production. So they're important crops, although they may not have the huge economic impact as some of the larger grains. These vegetatively propagated collections offer a lot of novel diversity, especially in the NPGS that you may not be able to find in other gene bank collections. They usually don't have an associated international center. So the US is often the place where people go to get information as well as germplasm for these collections. The materials are difficult or could be impossible to transfer across international or even some state borders. So in that case, it makes our collections particularly valuable and they may not be able to be replaced easily. In many cases, we have limited numbers of affiliated or associated research and breeding programs. So we may not have a large group of customers that are out there for each of these particular crops. But in total, there's a lot of these specialty crops we're talking about. They can be highly susceptible to abiotic and biotic threats, particularly because they are in a field or in a greenhouse location. They're usually inadequately secured at a secondary location, so they're pretty vulnerable. And often there's not as many technical resources available for these crops. We may not have easy propagation methods, especially for the crop wild relatives of these crops. We may not have in vitro tissue culture methods or other ways that make it easy for us to conserve them. Here's a map of the United States that has the plant germplasm system on it. And what I really wanted to emphasize here is that the red spots for the most part are considered the quote unquote, the clonal collection sites, with Geneva also being one of those. But the blue spots are additional locations that all have clonal collections associated with them. So although we may think of the clonal sites as being a handful of the NPGS sites, there's clonal collections almost throughout the entire system. There just may be a couple crops here and there for some of the locations, but the process of protecting and conserving these clonal crops is pretty much system-wide. And the concerns we have about them are system-wide. And the black spots on here are those sites that, as far as I know, are strictly seed collection sites. So the NPGS has almost 600,000 accessions in it, and they represent about 2,500 different genera. It's a really diverse collection. Of that, most of it, about 93%, is considered to be propagated by seeds. And that leaves about 7% of the collection, or 40,000 accessions, which is still a lot of accessions, that fall into this clonally propagated crop category. If we look at the fruits and nuts portion of that, there's about 69 different crops, and there's almost 27,000 accessions that fall into these 69 crops. Vegetables are a few of them are clonally propagated with about 5 crops and around 2,500 accessions. We have 9 grasses with 871 accessions. There's a group that I call the other category, the cacao, hops, rubber, and others, with 6 crops and 722 accessions, as well as a lot of ornamentals with 74 genera and over 9,500 accessions, primarily at the National Arboretum. These clonal collections are really difficult to manage. They're vulnerable. As I said before, they can be in the field, greenhouse, or in tissue culture. They usually require some sort of maintenance year-round. They're susceptible to pests and diseases. They're vulnerable to those weather events that could happen at different locations. They require periodic repropagation, whether it's annually for garlic or longer than that for some of the tree crops. We have a huge range in diversity, and the response of our crops to these, the range of that diversity responding to the abiotic and biotic stresses. We've got a wide range of phenologies and growth habits, rootstock responses, and all kinds of considerations that have to go into place as the maintenance occurs. And they get distributed in a wide range of different types of propagules across the different seasons. Across the bottom here, I just have a few of the threats that are affecting our collections these days. So it's important to secure these clonal collections. They're valuable collections, but it's also expensive and critical to make sure they're backed up. One way we could do that is by duplicating them, by putting them at a secondary location. We could also take some seeds from some of the wild species representatives and try to store them as seeds. We may put things into reduced temperature storage and tissue culture, or what we primarily focus on at our facility for the clonal types is cryopreservation. So here's a picture of our building at the NLGRP, and within there are about 82% of the NPGS seed accession secured, which is pretty good. Our challenge is that only 15% of those 40,000 clonal accessions are secured. So the backup is not nearly as good for the clonal collections as it is for the seed crops. Most of those clonal collections would have to go into liquid nitrogen, as shown in some of these liquid nitrogen tanks on the lower left in our vault. They could either go into liquid nitrogen, liquid phase, or into vapor phase. And they can be stored as shoot tips, dormant buds, embryonic axes. Sometimes seeds go into liquid nitrogen as well as pollen. We also have a floor for minus 18 degrees storage in the freezers, and that's for the seeds. We have a program at NLGRP for finding new ways to cryopreserve these clonally propagated crops, as well as implementing those methods for our collections. And I'll talk a little bit more about this in the second part of my presentation, but we can work with dormant buds, or we can take shoot tips excised from greenhouse grown plants, or we can take shoot tips excised from tissue culture plants. And we can use all of those different types as our starting material for cryo. We have a lot of different crops cryopreserved at NLGRP. And so we have methods for all these different crops, and they've been processed to different extents, depending on the crop and the technical resources. And this has been going on for over 30 years now at our facility. So some of these crops have actually been in storage for quite a long time. It's difficult to cryopreserve these clonal collections. It takes specialized facilities. It takes a lot of specialized skills and training. It's labor intensive. Ideally, a technician can cryopreserve about 60 to 75 accessions if they're having to excise out the shoot tips. And if they're processing dormant buds, they may be able to cryopreserve 200 or more accessions per year. But there aren't a lot of crops that are amenable to the dormant bud system. We need to make sure we have sufficient plant material, meaning we need to have the right amount, we need to have the right quality, the right time of year, and we want to make sure it's clean and free of endophytes or other contamination that may hinder the process. And we need to make sure we have tissue culture and cryopreservations available as well as tested, because often it's really difficult to repeat things in the literature without having practiced in your own lab. So this is going to conclude the first part of my presentation. And I'd just like to reiterate that the NPGS has a lot of vegetatively propagated collections throughout the distributed gene bank locations. These collections are really expensive to maintain. They're really vulnerable, but they're also really important. And we're going to have to think of creative, novel backup strategies to preserve these genetic resources now and into the future. Okay, thank you. Okay, part 2 is going to be about the preservation process of clonally propagated materials at the NLGRP. And this is a project that both Maria Jenderick, who's a plant physiologist and our collection curator, as well as myself, the plant physiologist, are involved in. And you can have a link down there at the bottom of the page for our unit's website that has some information about processing and materials at it. So the big picture that I'd like to talk about in this section is how do we go about the process of selecting materials, receiving, cryopreserving, making sure it's alive, and documenting that and who's involved in each of these processes. So this is my general flow chart. My next chart is the one I'm going to carry throughout this next section. And it kind of includes who are the people involved in making these decisions and what conversations are taking place. So the first step is identifying which crops or what crop we're going to be focusing on for cryopreservation and which accessions of that crop should be the ones that enter into this process. It's a really difficult, time-consuming process, so we need to be super smart about how we make these decisions. This decision gets made through a combination and through a lot of conversations between members of the crop germplasm committee, who may provide information about the vulnerability of the crop or the value of the crop or of specific accessions to be processed. It could definitely involve administrators, either research leaders, center directors, national program staff, or others who have information about incoming threats and vulnerabilities that need to be addressed. It needs input from the curation team and whether they have materials to provide at the time it's needed, as well as which materials would be priorities. And also the NLGRP scientist who needs to coordinate the effort within their staff members who are going to be doing this processing. Once those accessions of crops are identified, materials have got to be sent to our facility, and those are going to be sent from the curation team and received by members at NLGRP and entered into our databases, and identifier numbers will be assigned. Then they'll be cryopreserved. Then we'll have to check their viability and make sure they're alive. Then we're going to document all that in GRIN. And then at the end, we're working on coming up with some systems for reporting so that anyone can find out what the status of their crops are. So the first part I'll talk about is that crop and accession identification. Crop cryopreservation priorities can be based on a number of things from this team of people who are making these decisions. We'll need to have information about whether there's a method available to cryopreserve it, whether we've got plant materials available and what type of materials we need, whether there's technical support and personnel to actually do this work, both at the collection side to collect the materials as well as at the NLGRP side to process them. We have to consider the vulnerability and the threats to that collection or things coming down the pipeline that we may not be threatened yet, but could be very much in the future, as well as those administrator recommendations. There's also a lot of considerations into into deciding which accessions we're going to cryopreserve because it's a lot of work and we want to be smart about what we put into this pipeline because it may actually take us quite a while to get those materials into the pipeline and back out again in the cryopreserved state. Some questions we want to think about is whether we can effectively back it up as seeds. There could be some wild species in our collections that store really well as seeds that we can collect seeds from, and perhaps that's the propagule we should actually be storing rather than clones. And that would be great if that's possible. Another consideration is if it's already secured at a secondary location. If you've got part of a collection secured elsewhere, then maybe we want to think about preserving the materials that aren't backed up anywhere else first, just because they may be more vulnerable. It's just a thought. Another consideration is we want to cross-check our lists to make sure it isn't already at NLGRP. Some of these things could have been processed 25 years ago, and folks could have lost track of where they are in the pipeline after all this time, and how frequent we're going to be requiring new materials to be processing. We need to consider whether it's a taxon that can be cryopreserved using the available procedures. Sometimes we may have methods for the domesticated types of the species, or some things that people have worked with frequently, but then those methods aren't necessarily applicable to some of the wild species or to even some of the cultivars. So we need to be kind of aware of how widely applicable our methods are and which things we should be focusing on at this time. And we should probably be thinking about prioritizing accessions that are threatened in the living collections, such as they're difficult to propagate or maintain. They could be susceptible to abiotic or biotic stresses, and they could just be, quote unquote, the problem children. Maybe we want to make sure those are backed up. We also want to think about cryopreserving things that offer unique novelty to the collection, based on all that passport and genotype and phenotypic data we've been collecting. Perhaps we want to focus on things that are irreplaceable, something that came into the country decades ago, and we know we don't have access to that germplasm anymore. We want to consider what the priorities are for the CGCs and our user communities, our customers. We want to even take into account information we have about other collections, either within the country or elsewhere, and whether we've got something that's super unique and unusual versus something that's kind of widely available. We want to make sure we have healthy material available in adequate quantities and quality. And ideally, we need to have it in a form that we can easily cryopreserve. In a perfect world, we'd receive clean in vitro cultures that are healthy and easily propagated, and we can cryopreserve those. But the reality is that type of form may not be available for most of our crops. Moving on to the next part. We've decided what crops we're going to send. We know what accessions we're going to send, and now we're going to ship and receive them at NLGRP. So there's a decision and a discussion that takes place between the curation team and the NLGRP SY to determine how many accessions and how much of those accessions are going to get shipped and when they're going to get shipped. And we need to take into account when the optimal time to process this material is, because it's going to vary with every crop. Then when it's time to ship that material, the curation team emails the NLGRP SY and perhaps the cryotechnician with the date saying, we're putting it in the mail, expect it tomorrow, so we can watch for it. And ideally, they've got information about what the Green Global Order number was, or they've attached a spreadsheet that has the list of the accessions that are coming in the box. So now we're ready when that order arrives at NLGRP to confirm that it's complete, or whether it matches what's supposed to be in the box. And then an email can be sent to the curation team to say, yep, we've got it. And another email is going to get sent to our database team at NLGRP to confirm that we've received the order and what the order number was. And then we're also going to let them know what the cryopreservation method we're going to use to process that material is. Because the database folks are the ones that are assigned our NSSL inventory numbers. So it comes here from an inventory at the active sites, and then we need to have an NSSL inventory number so that we can process it and track it in our building. And I've got a page about that next. And then that database team also provides an eCryo card. And that eCryo card is kind of our data tracking method. It's a spreadsheet that has it pre-filled information about what just arrived, about the accession, maybe it's the cultivar name, inventory numbers, and those NSSL inventory numbers, so that we can track that in our labs. Here's just a couple slides about these details about what we were receiving. So an NSSL inventory number is for these clonal samples, and it's a 6-digit number that starts with NSSL. NSSL is because that was our former name. We used to be called the NSSL, which was the National Seed Storage Lab, until about, 15 to 20 years ago when we switched our name to NLGRP. It's a 6-digit inventory code. which are that 1 through 6 in right after the NSSL. And then there's a 2-digit code after that. And that 2-digit code has an 8, and that means it's going to be stored in liquid nitrogen at our facility. And the 1 means it's the first time we've processed this. So if you've shipped this thing multiple times, that number after the 8 could be a different replicate number from multiple processings. And then there's a 2-digit letter code after the 8-1 that tells what the inventory type is. And this is why we had to tell the data technicians, this is going to get cryopreserved as a meristem, or a BD would be a dormant bud. If it's just going to be stored in vitro, it would be an IV. So we have different ways in which we can keep track of what that sample looks like and what form it's surviving in while it's at our facility. The next part is about the cryo cards and the associated information for that. These are standardized spreadsheets that Maria's group and my group have been working on quite a bit in 2020 to come to a consensus about what information we want to have stored and what we're going to put into GRIN and how we're going to record that information. In some cases, we also may take images, and we can take those images and upload those to GRIN Global. And we're also now filling out standard templates for recording how we cryopreserve things and how we assess viability of those materials, so that, especially in the future, if somebody actually has to thaw out something that we've done and they're thawing it out 30 years from now, they want to know exactly what we did and how we recommend they be warmed so that we can actually get the things out of cryo successfully. So now these filled out templates for each crop are getting uploaded into GRIN Global and being stored as part of our databases. Okay, sample preparation and cryopreservation. So we can accept a number of different propagule types. We can use dormant buds in the wintertime for things like apple and willow. And there's a lot of research going on to determine other types of crops that this method may work for. And in this case, we're taking usually January harvested budwood from the field directly. Quite a few can be processed per technician in a season, but it's limited to the winter season. And right now, there aren't a lot of cold, hardy, temperate crops that this method works for. Hopefully that'll change in the future. We can also use non-dormant field or greenhouse plant materials. And great examples of this would be citrus or garlic, where we receive enough material to cryopreserve directly from the location. So we get a bundle of 200 buds or twigs with 200 buds of citrus, or enough garlic bulbs to cryopreserve 200 shoot tips of garlic dissected out of those cloves. Shoot tips get excised from them, and then there's adequate quantities provided by the active sites directly. We don't have to mess with the tissue culture prior to freezing. We do need tissue culture to regrow because we can't grow a 1 millimeter shoot tip into a plant any other way. So we do need to have some well-defined media. And this takes quite a bit of work. Hopefully we can do 40 to 60 accessions per skilled technician per year. There's a lot of seasonality to this process because we're taking those materials directly from a living plant and that needs to be at a stage that's amenable to the procedures. And then the third type of material we can accept are in vitro cultures. Strawberry, mint, banana, there's lots of examples of this. In this case, we'll get some tissue culture materials from the active site, or we can get material that we have to introduce into tissue culture first. And then we propagate that, we multiply it up so that we have enough shoot tips, and then we can cryopreserve those. There's always a challenge of endophytic contamination that could occur or may occur reveal itself during the process of cryopreservation, and that makes our jobs just a lot harder. And that's one of the big challenges of working with this tissue culture materials to start with. This is just my very generalized diagram of what happens when we cryopreserve. If we take dormant buds, they're going to get cut in, the twigs will arrive, they'll get cut into nodal sections. Those nodal sections will be placed in the cold and dried, dehydrated down to 25 to 30% moisture content. Then they're packaged into tubes and placed into controlled rate freezers where they're dropped down to about minus 30 degrees Celsius. And from there, they're put into liquid nitrogen vapor. Alternatively, if we're working with the shoot tips, we may be getting in materials directly from the field where we have to excise those, take some sections with the buds on them, surface sterilize them, or we could excise shoot tips directly from tissue culture materials. We've got a 1 millimeter shoot tip. They're tiny. It's really sensitive work to get those shoot tips excised appropriately and consistently from the right stage of the material. Then they're treated with cryoprotectants to remove the water and to replace it with chemicals that'll help it survive liquid nitrogen. They are then mounted onto foil strips with 10 per foil strip, and then plunged into liquid nitrogen and loaded into cryovials. Those cryovials are then put onto cryocanes, as shown on the right-hand side here. Everything's loaded into boxes and placed into a temporary liquid nitrogen holding tank until we identify what the viability levels are. So now we're on to viability testing. Viability testing can be done either at NLGRP or at the active site, depending on what method we're using. For the dormant buds, our preferred choice is to graft it, because then we've got a living tree that's propagatable, that's ready to be planted in the field if they wanted to. And that is ideally done at the active site because we don't have those resources necessarily to NLGRP to do all that grafting. An alternative of something we can do at NLGRP is to force buds, so we can get an indication as to whether a bud will form a shoot, but we don't always have, or we don't have data that correlates the bud's ability to form a shoot through forced bud grafting and its ability to be grafted onto a rootstock. So there's some research in progress right there. Another thing we can do is take the shoot that forms after forced bud development and excise out a shoot tip from that, sterilize it, put it onto medium, and then grow it in tissue culture. And now we can have a tissue culture plant, which is something that we can propagate and move to the greenhouse if we need to. Alternatively, we can pull something out of liquid nitrogen, and if it's a shoot tip, then ideally we can just plate it, we can warm it, we can dilute out the cryoprotectants, and then we can plate it onto medium. And then we can grow some plants. And those plants can root, and then we can have a viability through the regrowth levels we get in tissue culture. In the case of citrus, we were not able to get those shoot tips to regrow directly by plating them onto media. So we went through a micrografting process where we start up in vitro rootstocks, and then we put our shoot tips onto the in vitro rootstocks and grow them onto into plants at NLGRP. So now we're going to collect the data for those successions. And the NLGRP usual standard is meaning it's able to regenerate a plant and a total of 60 predicted viable explants that are cryopreserved. So we call that our 40-60 standard. If we meet that standard or something else that we've decided on is appropriate, then those samples that are cryopreserved can move from the temporary tanks into our vault, and we'll record the information about where it went into the vault, which cryotank, as well as its viability data and storage locations, and how it got cryopreserved into that eCryo card. Within the vault, everything is labeled by these cans, which have these lids, and the lid has a barcode and some information on it that says what tank it's in, which section of the tank it's in, and then what box it is in. So here's a cool diagram that, that Kat Chen made that shows the 6 sections of a cryotank. You're looking head on into it. It's a big circle. It's on a lazy Susan. And within each one of those squares, that's a box. And inside that box has these cryocanes of cryopreserved materials in it, or the tubes of the dormant buds. So there's 6 different sections, each with a different color code on it. And we know where each sample is based on its label and its code, and that information is entered into GRIN. So moving on to GRIN global documentation. So that eCryo card is filled out by the technician, as well as any associated images, and the preservation and viability methods are all provided to the NLGRP database team. That database team that enters the data into GRIN Global and sends a confirmation email to the NLGRP scientist and cryotechnician to say, yes, it's done. Your data is entered into GRIN and we can consider those backed up at NLGRP. The last part is this cryopreservation reports. It's not something that's been offered in the past, but it's a new project we've got going on with Cullen McGovern, and he's part of that database unit here at our facility. And he's making some automated reports where we can select and download crop preservation data from NLGRP when logged into the GRIN Global public website. So you don't even have to be part of the GRIN Curator system. You can just log into the GRIN Global public website and pick a report, and you can search on taxa, identifier numbers, dates into liquid nitrogen, active sites, inventory types, and more. And ideally, you'll click on that and you can get an Excel spreadsheet that has the information that you need. So we are working on doing that right now. Part of the problem is that when you start getting these nice reports out, you start to see all the inconsistent recording issues we have over the past 30 years. So there's also an effort to clean up some of these things within GRIN so that we have the data in GRIN reported consistently so those reports actually make sense. There's a lot of information about cryopreservation that I didn't talk about today, but I can use this as an opportunity to advertise about our training resources. And I have an ebook that we are editing. And there's a few chapters that are public right now. There's a bunch of them that are hidden that will be released very soon. And it actually goes into the nitty-gritty details about how to excise shoot tips, how to cryopreserve different types of crops, both dormant buds and shoot tips. So if you want more information about what actually goes on in the cryo part, I recommend you take a look at this ebook. I also put down 2 references here, and these 2 references are some review papers we published last year about dormant buds and about shoot tip cryopreservation that actually talks through each of the processes and the challenges involved with each of those steps. And then I'm just going to mention some of the staff members. I probably have even missed some that are involved in clonal cryopreservation and data management here in Fort Collins. My team includes Rami and Ashley and Brad and Adam, as well as Kat Chen, who made all these great diagrams and illustrations in my presentation today, or most of them, and Emma. Maria's group's permanent staff members are Diane, Elise, and Barb Ambers. And Stephanie's group is Renee, Amelia, and Colin. Part 3 of my presentation is about this online learning and this plant genetic resources management and use project we have going on. It could easily become a full-time job to assimilate all the information that's out there and build it into this online content that we want to share. Our goal is to assimilate information about both genetic resources conservation as well as their use, and make that available freely through GRIN Global, or through GRIN. And we're doing this in collaboration with the database group in Beltsville. There's also going to be some online classes starting next fall. The first 1-credit class is going to get offered through Colorado State University. And the project right now is funded by a NIFR Higher Education Challenge Grant as a collaboration between CSU, Iowa State, and USDA. The information that we're generating in this program is temporarily available at this website at the bottom. We needed a holding place for public content while we work on developing the GRIN site. The idea is the GRIN site will be available sometime this spring. So we have a lot of topics, and we're trying to figure out how we're going to organize this content right now. And my current list of training topic categories are introduction, basic information about plant genetic resources conservation, aspects of collection maintenance, which is a very broad topic, including seeds, clonals, pathology, regeneration, distribution, backup type content. We'll have a section on characterization and evaluation, so genotyping and phenotyping, documentation and GRIN Global. And then we go into more of the use side, which would be crop wild relatives and domestication, plant breeding. We have virtual tours in the pipeline, as well as information about crops and cultural diversity. Training and learning resource content is going to come available in many different formats, such as ebooks, courses, online classes, webinars, videos, infographics, and online resources. And in fact, Kim and the seminar speakers as part of this webinar series are all are agreeing to let me flip the content of these webinars into training resources also. We have 3 ebooks that are available publicly right now. This includes one on crop wild relatives and their use in plant breeding. There's one that I already spoke about, of the cryopreservation of clonal propagules. And then we did a field tour of the USDA National Collection in Davis, California. So if you ever miss John Priest, now that he's retired, you can go to our ebook and there's 10 videos of John talking about all the different collections out in the field and telling his stories. And these are all freely available now. You can go to our website and take a peek. We've also been making infographics. So you've probably seen these already. They've been distributed by program staff. We have one about conserving genetic resources, and we have one about breeding and the use of genetic resources. They've been translated into 6 languages, and we have more infographics planned. There's one in the works for the role of botanic gardens in plant genetic resource conservation. And then I want to work with the outreach committee of the PGOC to do one specifically about the NPGS. Again, you can get all this information and use these for any purposes from our temporary website. This is just the banner for the future website that's under development associated with GRIN, saying that yes, it's going to exist. It's just not quite out there yet. There's a lot of behind the scenes works that the database management group is doing right now for us. So there's a lot of content that's in the works. And the next training resources that I'm focusing on are developing some ebooks based on gene banking principles, addressing pathology issues in gene banks. We have our first chapter that we'll be releasing hopefully very soon. An ebook about the genetics and genomics of gene banking, another one on phenotyping collections. We're working on an NLGRP virtual tour, which we'll share, as well as content relating to crop and cultural diversity. We know that we are not covering everything yet. So my list of things I need to work on, but I've kind of got a lot on my plate right now, also include things like seed conservation and storage, regeneration, distribution, and documentation. So this just lists the projects, team members for our Higher Education Challenge Grant. It's coordinated by Pat Byrne, who's retired from Colorado State University, but still working, seems like nearly full-time on this effort, as well as a number of researchers and students at CSU, Iowa State, as well as some members of USD. So that concludes my presentation. I actually came within the time limit. I'm quite thrilled, and I'm open to questions.
Okay, Gayle, thank you very much. I suppose, do you want to, yes, let's give Gayle a round of applause here. That's wonderful. Let's see, let me start my video. I don't see any questions in the chat room, but anyone is invited to put a question in the chat there. Are there some people online that have a question for Gayle at this point?
I do.
Sure.
Great.
Hi, Gayle. Hi, amazing work. My goodness. So, Gayle, you gave the numbers that can be cryopreserved per full-time technician. How many technicians do you guys have?
You saw the whole list. There's not very many. So
there aren't very many.
Probably spends half their time of total hours on cryo efforts right now. The rest of my staff's time is spent on all of our genetics work, which I did not even touch on today, as well as all these learning resources that we're developing. So we're doing that in-house with staff members too. So I probably have the equivalent of 2 full-time staff members that are working on training or that are working on the cryo. And Maria's group is full-time cryo, and they've got 3 staff members right now, plus some perhaps temporaries.
So do you think you have enough
Which means
resources?
the reality of getting through all this work we have to do
Yeah.
It's really hard. And that's the scary part, because the job
Yeah.
is kind of impossible given the resources that are available and the fact that we don't have methods available for a lot of these crops yet. So there's a lot of research that has to happen behind the scenes.
Yeah. So what do you think you need so that you can get your assignment done?
I think
I don't know if it's a good question to ask, but
Nahala, she
I
needs,
think we need to be really smart. I think instead of saying, I need more resources, I need more, we need to be really smart on how we approach the questions.
yeah.
And I wanted to spend some time at the front end on when folks send us materials, be super smart about what you're sending and make sure it's your highest priorities, because resources are limited. And yes, if it's the citrus collection and it's super high risk and problems are around the corner, then it jumps to the front of the line.
Right.
In other cases, and in citrus, actually, the method got developed through grant writing. So in collaboration with Riverside, we were writing grants and we had some soft funds to develop the methods.
Yes.
What we're running into is that soft funders or granting agencies are not that excited about processing. They like technologies. So we can get grant money to develop a technology. It's a lot harder to get a grant money to say, now I'm going to process 50 a year.
Right,
and that's really boring. And so that's where we run into challenges. And that's where we need to start being super smart about, if I can only do 50, which 50 am I going to do first?
Yeah.
So that's part of the thought process that we need to be a team as. And that's why I actually picked this topic today to talk about, because it's not just us making the decision, and it's not just the curation team making the decision. It needs to be this big group discussion.
Right.
so
Well,
Gayle,
thank you for everything you do.
thank you, Nahala. Ben Gutierrez has a question. How do you balance the challenging crops with the easy ones like apple? And then do you budget a certain amount of time for each crop?
That's a tough question, because I don't even think apple's that easy. You can put air quotes around easy, because yes, dormant buds of apple are probably the easiest thing we have, but the dormant bud method we have for apple doesn't work very well on all those crop wild relatives you have, like the wild species. So a lot of those, if you start getting into the data, they weren't that amenable. And was it because the rootstocks didn't work real well for the viability testing, or was it the cryopreservation method? We don't even have those answers. I'm all about personally doing the easy stuff. If like we're doing apple this year because you've got some super vulnerable materials that we need to just get in, and relatively speaking, that's going to go in easily. We've also spent quite a bit of time in my group this past year working with Kim's group to cryopreserve materials that they had already put into tissue culture. So they put a tremendous amount of work to put pears and mint and strawberries and hazelnuts and other crops into tissue culture over the past 20 years. And some of those were no longer going to get maintained in tissue culture. So the thought of having all that tissue culture material completely available and methods available, to me said, we should just go ahead and cryopreserve this stuff, because the amount of work it's going to take to put all that back into cryo again, or to put into tissue culture again, is tremendous. So we better take advantage of the resources we have while we have them. Other times we've brought things like citrus to the front of the line because it was threatened and we had to do something fast. Unfortunately, everything is not a success story. We all have our crops that were challenges where we spent a lot of time on them, and yes, they're super vulnerable, but it just didn't come together. And we tried, and they were research projects that never, for many different reasons, weren't as successful as we wanted them to be. And in those cases, we had to work back with program staff and curation teams to say, let's think out of the box. You can't just send it to NLGRP and solve the problem. Maybe we need to find a duplicate collection somewhere else. Maybe we need to find a way to secure this collection before it's lost, because it's not possible with the technologies we have and the resources we have at this time. So it's a constant balancing game. It's actually a really hard part of the job.
Gayle, I have a question. I think that when Dr. Barbara Reed started at our unit back in the late 80s, 1989, her first project was cryogenics, well, tissue culture and cryogenics of strawberry. And I'm guessing that a number of the strawberry shoot tips that she initially put in to cryo went to your location, and they're now there's, you know, decades of material. Do you guys have, are you checking viability? Do you have plans for regeneration, or is once in cryo, always in cryo? What's the procedure?
Good question. Maria has some long-term storage experiments. So she has some projects going on that have probably been going on for 10 or more years now. And they may even be pulling out some things from the older collections to confirm that the viability is still good. When we first started our citrus project, we set up some viability assessments that we're doing periodic checks, but we purposefully, and same thing with Maria's projects of the longevity, they were purposefully cryopreserved to a larger extent. So we've got a lot more accessions that are expendable to be checking viability over time. In other cases, like the apple, they're getting pulled out quite frequently because there are fire blight issues in the Geneva collection, and they actually use their cryopreserved materials. So we get informal confirmation that things are still doing fine because we're actually pulling them out and using them. With respect to Barbara Reed's older materials, the trick is that a lot of hers, she only has maybe 3 or 4 vials each. And she was putting 25 shoot tips per vial. So she put tons of shoot tips in a vial, but we don't have very many vials. So if we go in and do a viability check, we just took 1/3 of the collection away. And that's a scary thought. So we're trying to do most of these viability checks on accessions for which we have quite a bit of material, because we don't want to deplete our accessions confirming that it's still alive and then realizing we have nothing and we have to do it again.
So once you take it out to sample, that whole quantum of material is used.
It goes together. It's a one-shot deal. Yeah.
Oh,
And that's why things got switched from having 25 per vial. At one point, Apple had more as well per vial or per container. And now we're down to 10 each, because 10 gives us, if we have our 40% viability requirement, then statistically, we have extremely high odds that one of them will be living out of that 10. And that's all we need for our clone. So we're happy if one survives. And it also gives us more chances for replicates. So in shoot tip world, and ideally in dormant bud world, we have 15 tubes of 10 each. So we can do multiple regeneration events as needed in the future. So hopefully we can cryopreserve the stuff once and really not have to look at it again anytime soon. Because we don't want to spend our time repeating all that again when we have so much that we haven't even started yet. And something's better than nothing.
Hi, Gayle. I loved it. Thank you. We love our Fort Collins friends. I mean, you are a lifesaver. We love the effort that you put. We do use the collection for in cryo a lot. And I had a question on those lines about, you know, so we'll look at our list, what has died in the field, and we'll hit an accession that there's a single tube left in cryo. And so really the only genotype potentially available is this thing in cryopreservation. So who owns that in theory? And, you know, and what sorts of situations do we ever risk pulling that thing out to test it, to maybe start repropagating it?
Technically it has an NSSL number on it, but it's an accession that is associated with Geneva. I think it's a joint decision. If you're ready to take that plunge and take that last accession, it's all yours. NLGRP clonal programs serve the active sites. We are working in collaboration with you. Nothing that comes to NLGRP is given to anyone other than an active site. So if a collection, whatever, if a university called us up and said, can I have this? We'll say, no, you need to talk to Ben to get that. You're not coming through us to get that material. We do have some research projects that are collaborations with other organizations, but things that are part of these backup programs are directly associated with the active site, and we consider the active sites to be our customers.
Thanks, Gayle. That's a lot of pressure on me and the rest of the curious.
Exactly. So be really, really ready when you ask for that last accession.
Yeah.
Well, thank you. Nahala, I think I cut you off a little bit. Did you have another question?
No, I'm good. No, thank you.
Okay. Gayle, in your talk, you said something about if I know some of us sites have set up backups at other locations. For example, the hazelnuts are stored at Parlier. And yet when program leaders came by, and it's sort of like, it's no countsies unless it's at NLGRP. So, but still, you'd like us to, in our prioritization, if we do have certain things backed up at another site, consider that may knock down that priority and take things that aren't backed up. Is that what I'm hearing you say?
Yeah, that's what I'm saying. It should just be a consideration. And our challenge is that even with the NLGRP numbers, we don't always meet our own standards. If Ben's asked for a lot of these apples already, now these things that used to be cryopreserved to the standards we want no longer meet our standards. So it's always a game in flux in terms of, are we securely backed up at NLGRP, and what's the definition of a backup anyway? And this came up a little bit at the PGOC meeting last June, when I believe it was Chris Walters mentioned the differences between backups, which may mean security backups, which may mean we meet the standards and we can check this off the list. Or maybe it's a duplicate. Maybe it's something that we cryopreserved, like in citrus. We cryopreserved it. It doesn't quite meet standards. Maybe we didn't have enough material. Maybe it wasn't quite high enough viability. But you know, we're going to keep it anyway because it's better than nothing. And better than nothing is important, because if you're down to the last tree in the field and something happens, then wouldn't it be nice to have something rather than nothing? So there's actually a lot of questions, and I keep bugging people about wanting to be involved when there's a discussion about what that checkoff box in GRIN means when it says backed up, because I don't even know what that means. And I think the site is in charge of that box, but I think everybody uses that box differently and some don't use it at all. And I think that would be actually a great discussion at some point amongst clonal curation people, including those at the locations that aren't necessarily considered clonal sites, on how they use the word backup. And it's a question for the seed universe also. What is a backup? And what's the difference between something that's a security backup that's officially done and crossed off the list, or a duplicate, which is better than nothing? And when do we start reprocessing the duplicates, like Ben's last tubes, versus saying, I got to get the new stuff in? Those duplicates are better than nothing, and we need to move on. So there's a lot of questions out there, and that's a huge gray area. And I think those are discussions that need to be had.
Yes, that piqued my brain there a little bit. So where we have just done a distribution sample to you, and you guys are in the process of backing up because we sent some germplasm to you in one form or another, maybe seed, maybe tissue cultures. So you're saying it's us that go and check that box that says NLGRP backup, or I think it's NSSL backup, but
I don't even know. I actually, if I say something, I could be wrong about that. From when I see that data downloaded in GRIN or whatever, I'm never quite sure what that box means. And I don't think that's been well defined. And I think it's more used on the seed side than it is in the clonal side. But some sites I don't think use it at all. So I think it's very variably used, and I think some definitions would be a good thing. And I'm actually not quite sure who's in charge of it.
If I could just have a quick, I'm going to share my screen. Is it coming in?
That's the one. It's that it's backed up question.
Yeah, so it says NSSL is the backup. And I guess that I could edit that yeah, so I see that I could add that, but so that's up to me, not you guys. You don't mess with that.
I don't think, I know it may be that NSSL location. I don't think that's us. I could be wrong. I shouldn't say. It would have to be our database folks. I think Cullen's on the line. He may have an answer to that, but he may not know either.
Yeah.
the
Colin,
carriers own the accession, but the NSSL would own the inventory, right? So the backed up option is under the accession. So it really, the site should be the only one that has that permission, right, to change or update it.
That's what I think. And our NSSL information is in a different table. And that's where you have the information about the tank and the backup and the viability and all of that. That's elsewhere. I think this is your inventory table.
so then when Peter makes or wants us to report, it's your files is where he goes, not this other NSSL location one backup that we don't count from that. We count from your files.
I don't know. I don't know if that's consistently done. Hopefully these reports that Colin is helping us develop will give you the up-to-date information, and it'll actually compile, it pulls it from all the relevant tables because we're storing our data in a number of different tables.
Just briefly, I can answer that a little bit.
Yes, please. Thank you, Colin.
So I don't, that field that you just showed me is new, and I'm surprised that you can edit it. If I were to count the number of inventories that we had backed up for a certain site, I would look at our NSSL prefix inventory, and it would have a field that links directly to your inventory, which must exist and is linked to you. So then I would say, tell me which site this person works at who owns this inventory. So the way I would count would be quite different, but I think more accurate.
Well, I guess what I showed was at the accession level. And of course, I'm trying to get the data here at the moment. It's taking its little circular route. I was gonna go to the inventory item where your guys' inventory links up, or if I open the inventory for that particular number, yours is present and ours is present. At least I can see yours. I can't edit yours.
Right.
But so you would take it from that inventory count then of your own inventory.
Yes, I would. And if we had some firm conditions about what backed up means, I would only count it if it met those conditions.
There's a lot of NSSL numbers that don't have data associated with them. So things that got sent to NLGRP in the past that never actually made it through the cryo process for whatever reason. And those NSSL numbers very well might still be hanging in there with no data associated. So you can't just say if it's got an NSSL number, it counts. Like Colin said, it has to meet criteria such as having whatever those criteria are. It has something in liquid nitrogen, and that something has an associated viability.
Okay.
And so that's what you're pulling from different tables. And that's why these reports are going to be so handy, because then you can get your strawberry report or your Corvallis report, and you'll have that information all compiled in a spreadsheet.
I see some comments in our comment field, and I don't mean to monopolize your time here, Gayle. Jenny has a comment. Perhaps this would be a good topic for Marty to tackle in his GG Q&A sessions. And it seems like this is an area where we all could use some training when and probably how consistently to add the information to the inventory table. Yeah, because it sounds like it's not necessarily been done consistently over time.
And I think before even Marty does that training, I think we need to have people come together to decide what the criteria are. And what do we want done there, and how do we want that recorded? Because I don't think he has the answer to that either, at least as far as I've been able to find out. I don't think there are necessarily standards on the clonal side. Maybe there's something for seeds, but I've never seen something written and codified for clonal to say, when do you get to check that box? And who gets to check it? Thank you, Jenny. I think it'd be great if you could bring it up at AdCom meeting. And please let me know when that happens. Any others? I think we're about done, but feel free to contact me anytime. I'm always around and I'm happy to answer questions. My plan is to take at least my first two quote unquote presentations and turn them into training content and then post them as webinars up on our training content website at some point. And I'm asking permission for each of the speakers for the series to do that. So all that information will be flipped and made public.
Let's see, Robert. Oh, he also compliments you on your presentation.
And Kat did a beautiful job on those illustrations. I'll call her out again.
Thank you so much for a great presentation, Gayle. You have opened up some questions that we need to address, definitely. And we're really happy to have you have the time to come and present today. And I think this is a great moment that we can become more aware of the coordination we need to do as a system. So we'll look forward to some better interactions as we go forward. Maybe this is a suggestion we can put to the PGOC, because I know that Brian was asking just recently about items for PGOC coming up. So yeah.
Yeah.
Any other? Go ahead, Gayle.
Oh, it is a good idea.
Okay. Well, thank you so much, everybody. We will have one more in our series next week when Jeff Garstens will be giving our presentation at 1:30 Pacific time in a week. So it's great to see you all, and thanks very much. We'll talk to you in another week. Bye-bye, Tim and everybody.